In this video, we will discuss about E-Cadherin laws could also help the tumor development. We will first discuss about the normal functioning of the E-Cadherin. So E-Cadherin is usually expressed on the cell surface and it helps the cells to remain at its site. And how these interactions works is the two interactions of the E-Cadherin cell surface of the cells are shown here, where we can see that on the left figure on the right hand side you can see the one of the cell surface one of the cell cytoplasms, and on the left hand side we can see the another cytoplasms, and in between there is an intercellular space, and we can see that they are interacting with each other, and that will triggers the downstream intercellular signalings or downstream complex formation of P120 and alpha, beta, and gamma catenin. And what has been found in the literature is that with time or because of any reasons that there is a mutations occurs in any of these intercellular parts, for example in beta catenin, then it will cause the loss of the E-Cadherin functions. That is, the cells will not be able to interact, cells E-Cadherin will not be able to interact with the other cells E-Cadherins, and this might leads to the loss of these epithelial like characteristics, and these cells might acquire a mesenchymal like characteristics, which is one of the important characteristics for the tumor metastasis. And these things are shown in detail or slightly in detail in the figure on the right hand side, where we can see that the tumors or benign tumors are present within the tumor capsule, and at this stage they are not harmful to the patients since they are intact at its site and are not affecting other tumor cells. However, once and one of the properties of these benign tumors is that their epithelials are functioning normally or benign, or they are called as benign epithelial tumor or adenoma. And once the mutations occurs in a way that will lead to the loss of these E-Cadherin functions, then it will cause disruptions in the cell adhesions, and these tumors will now will categorized as malignant tumors, and their property is to migrate from one place to another place. So they will escape the tumor capsules and then migrates to a distant organ or nearby organ, and these things are being categorized as these metastatic tumor cells or the metastasis. And the stage at which this tumor starts to migrate from the tumor capsules, they are categorized as carcinoma. And so once the cells are benign, they are called adenoma. And once they acquire the characteristics of the uh, once they acquire the mesenchymal characteristics, they are categorized as carcinoma. And this is one of the reasons why these pancreatic cancer cells are also known as pancreatic adenocarcinoma, because they took these pancreatic cells took a long time to reach to this carcinoma stage, and uh, based on some mathematical model it shows that it can take around 10 to 10 to 15 years to reach from adenoma to carcinoma stage. And the reason is because the loss of E-Cadherins, one of the reasons, not this is the exact reason, one of the reason is the loss of E-Cadherin functions because of the mutations that occurs in the tumor cells. So this is uh, the one of the old paper that shows that is published in one of the old review paper that is published in 1999, that shows that E-Cadherin loss could also helps in the tumor development, and that can be evident from the it can be evident from the data that shows that mutations in the pancreatic cancer or the cancer patients, most of the some of the cancer patients also shows these mutations in the one of the intercellular components, and that might affect the loss of the E-Cadherin functions. So these mutations or disruptions in these intercellular parts can also be one of the indicators towards this carcinoma stage. So I hope you understood to some extent how this E-Cadherin loss could help the tumor development. So if you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.